Assalamu alaikum and a great day to everyone. Welcome to this session today with our speaker and special guest, Professor Dr. Robert Lustig. Today's session is part of a series of podcasts where we discuss the four pillars of health and wellness model of S.E.L.F, which I have created and which includes sleep, exercise, lifestyle, and food. Today, we will focus on food and health. And to discuss this important topic, we have as our guest Professor Dr. Robert Lustig, who is a trailblazer in the field of metabolic health. So thank you, Dr. Lustig. It's a pleasure to have you with us. And thank you very much for making time for this important podcast, especially for the audiences in this part of the world.
Thank you for having me, Dr. Ahmed. And by the way, I'm Rob, not I'm not your doctor, so I go by my given name. This problem.
So shall I introduce you first before we start?
Sure.
Yeah. So with your permission, for the audience, Dr. Robert Lustig is a professor emeritus of pediatrics in the Division of Endocrinology at UCSF, where he has been serving for over 23 years. Renowned for his nutrition and public health expertise, he graduated from MIT in 1976, received his MD from Cornell University Medical College in 1980, and completed a master's in the study of law from UC Hastings. He's the author of several influential books, including Fat Chance, Beating the Odds Against Sugar, Processed Food, Obesity, and Disease, which was a New York Times bestseller, Metabolical, which is my all-time favorite, The Lure and the Lies of the Processed Food, Nutrition, and Modern Medicine, The Hacking of the American Mind, The Science Behind the Corporate Takeover of Our Bodies and Brains. The next book is called The Fat Chance Cookbook. Then Sugar Has 56 Names, A Shopper's Guide, Obesity Before Birth. And there are plenty of other papers. So welcome again, Rob. And let's get started with our conversation, which I'm sure is going to be very informative, shifting our mindset and perspective about metabolic health and chronic diseases with a focus on food and diet. So welcome on board.
Very good.
So my first question obviously is, what is metabolic health and metabolic dysfunction? And what is mitochondrial dysfunction and insulin resistance, if you can start off with that?
So the first issue is what's metabolic health. So there are three words that get thrown around and they get confused quite a bit. So let's define them properly. One is food science, one is nutrition, and the third is metabolic health. So what's the difference between those? Food science is what happens to food between the ground and the mouth. Nutrition is what happens to food between the mouth and the cell. Metabolic health is what happens inside the cell to that food. That ultimately confers health, wellness, or disease. Now, metabolic health is clearly the most important. Metabolic health is the North Star of medicine.
Mm-hmm.
The problem is that nutrition can inform metabolic health, but it is one step divorced from it. Food science can inform metabolic health, but it is two steps divorced from it. So what we really need to know is what's going on inside the cell. And nutrition does not actually answer those questions. Adding up a column of numbers and saying, well, you meet the recommended dietary allowance means absolutely nothing. Because number one, you don't know if it got absorbed. Number two, you don't know if it got to the cell of interest. Number three, you don't know if it was metabolized. Number four, you don't know if it actually activated the transcription factors, receptors, or enzymes that were necessary. Number five, you don't know what the signal transduction systems were beyond that. So the bottom line is metabolic health accounts for all of that. But you have to know the science in order to be able to understand that. And unfortunately, this is not really taught in medical school. It is certainly not taught in dietary school. And so it behooves us to be able to, understand this and, convey this, continuing education to physicians so that they, can take this information and use it properly.
And what was the relationship of mitochondrial dysfunction linked with insulin resistance, if you may add on that?
Let's one step at a time. So the biggest issue in metabolic health is energy metabolism. Okay, we have a problem of obesity, we have a problem of diabetes, we have a problem of heart disease, we have a problem of fatty liver disease, we have cancer, dementia, we have polycystic ovarian syndrome. None of those have any cures. Not only that, but none of those have any genetic etiologies that are hard and fast. They're all considered, quote, polygenic. The fact of the matter is, genetics hasn't solved any of these. And the reason is because none of these diseases are genetic. They have to do with energy metabolism. And where does energy metabolism occur? It occurs in the mitochondria. So what are the mitochondria? The mitochondria are the little energy burning factories inside each cell. Depending on how active that cell is energy-wise will determine how many mitochondria there are. So for instance, the brain, which is the most energy-intensive organ, has the most mitochondria, muscle next, and adipose tissue, which is not particularly metabolically active, will have the least number of mitochondria. On the other hand, there are brown adipocytes as opposed to white adipocytes, and they burn energy like crazy. That's why they're brown, because mitochondria are brown. So mitochondria determine the rate of energy metabolism. So what do mitochondria do? They convert the chemical energy in food into the chemical energy that your body can use, called ATP. And the question is, at what efficiency, through what mechanism, and are there any diversions? And when I say diversions, are there toxins that inhibit mitochondrial function or even potentially stimulate mitochondrial function? Or are there signals going on inside the cell that will divert energy, say, from going all the way to carbon dioxide and ATP and possibly divert it off to fat, off to amino acids, off to ribose for DNA? The way to think about this is every cell in your body at one point in its life had to grow, and at another time it had to burn. Now, what determines whether a cell grows or burns? And the answer is three enzymes. Three enzymes inside the cell determine whether a cell grows or burns. Those three enzymes are called PI3 kinase, phosphatidylinositol 3 kinase. This is the spigot that lets the glucose into the cell in the first place. This is the enzyme that is problematic in cancer because cancer cells don't have mitochondria. Cancer cells don't need mitochondria. Otto Warburg won the Nobel Prize in 1931 for realizing that cancer cells don't need mitochondria or oxygen. They ferment. And from fermentation, they get enough ATP to be able to power the cell, and they're also able to use the byproducts to build new cells. So understanding how the cell takes that energy, that food energy, and ultimately diverts it either all the way to ATP and carbon dioxide, meaning mitochondrial function is maximal, or diversionary to these other pathways, the pentose phosphate shunt, the Randall cycle, de novo lipogenesis, to make other components, is essential. So PI3 kinase is the spigot that lets the glucose in the first place. The second enzyme that's important is AMP kinase. Now, this is the fuel gauge on the liver cell. So when your cell becomes ATP depleted, it's turning ATP into ADP. It's cutting off a phosphate bond, and that phosphate bond has the energy in it. The electrons go through the electron transport chain, driving ATP synthase. That's how you do it. So the ATP becomes ADP, and then the ADP becomes AMP, adenosine monophosphate. Well,
Yeah.
adenosine monophosphate then fits inside the active site of this enzyme called AMP kinase, and it tells that cell, hey, I need more mitochondria because I'm now energy depleted. Because if I got all the way down to AMP, I'm not doing very well. I'm way
Mm-hmm.
behind the eight ball. And so that will actually be the initiator of new mitochondrial function and also increased mitochondrial biogenesis and activity. So AMP kinase is essential in terms of driving all the way to carbon dioxide and ATP in order to make new molecules. Uh, the third enzyme that's important is called mTOR, mammalian target of rapamycin. And this is
Mm-hmm.
the enzyme that basically tells the cell whether it's time to divide or not, okay? Whether
Okay.
it's time to grow or not. Now, these three enzymes are all kinases. They are all activated by changes in phosphorylation status of the cell, which, of course, are changed by food. So when the food's coming in, the phosphorylation status of those three enzymes change so that the PI3 kinase goes up to bring more energy in. The AMP kinase goes down because you're going to use that energy for other things because you have enough energy. And the mTOR will be on because you might divide. When you're burning, like for instance, a muscle cell that's in, an exercise, okay, and you need all the ATP in order to make that muscle cell contract and relax and contract and relax. Okay, then you're going to do the exact opposite. You're going to bring that PI3 kinase down. You're going to bring the AMP kinase up. You're going to bring the mTOR down because this is not the time to grow. This is the time to burn.
Mm-hmm.
What we've learned is that growth and burning are the things that matter the most to a cell.
Mm-hmm.
And when those three enzymes are in synchrony, you will get one or the other. But what if they're dysynchronous, those three enzymes? After all, you have three enzymes. They have two valences on, off, or high, low, if you will. Okay, which means you have 8 permutations, 2 to the 3. So we've described 2 of them. The other 6 are all chronic metabolic disease. So the dysynchrony of these enzymes is what leads to inflammation. It's what leads to aging. It's what leads to energy storage. It's what leads to insulin resistance. The dysynchrony of these 3 enzymes. So what this means is that we need to try to keep our energy moving through the cell
Mm-hmm.
as efficiently as possible to keep those 3 enzymes doing the right thing. And what we also need to do is we need to try to reduce the uh, uh, drivers of diversion. And those drivers of diversion are reactive oxygen species, which mitochondria
Mm-hmm.
will make, and also uh, inhibitors of mitochondrial function, like, for instance, air pollution, like um, toxins, chemicals in the environment. And the big kahuna, the one that I rail against the most because it's the one we put there on purpose, fructose, because fructose is a mitochondrial toxin and it
Yeah.
inhibits the enzymes of the mitochondria so that it cannot generate ATP. Rather, it generates fat
Mm-hmm.
because it's diverting the energy to de novo lipogenesis, new fat making. It's turning sugar into fat. And this is the primary driver of chronic metabolic disease.
Yeah, great. So thank you very much for that. And at least I'm more educated about this. And so, is it fair to say that metabolic health is actually mitochondrial health?
Absolutely. Metabolic health and mitochondrial health are basically synonymous with each other.
Yeah.
And so
So,
understanding how mitochondria work, why they work, and what ultimately impacts their function is essential to understanding modern medicine and trying to ameliorate these chronic metabolic diseases. What we've learned is that none of these things in the mitochondria are druggable. There's no drug that gets to the mitochondria. They're all foodable. That is, the food gets to the mitochondria. That's their job. But unfortunately, some foods make the mitochondria work better, and some foods make the mitochondria work worse.
Mm-hmm.
And so you have to know which is which. And unfortunately, the food that we are all eating today, whether you're in the United States or in Pakistan or anywhere else really in the world, is this stuff called ultra processed food. Now, ultra processed food has gotten a bad name, and for good reason, because it's bad. And the reason is because ultra processed food is has many things wrong with it, but in particular, four things that impact on mitochondrial
Mm-hmm.
metabolism: too much sugar, too little fiber, too few omega-3 fatty acids, which are necessary to build mitochondria,
Mm-hmm.
and too many emulsifiers, which ultimately lead to inflammation, which impair mitochondrial function as well. So, ultra processed food is a problem. Ultra processed food damages our mitochondria. Ultra processed food drives chronic metabolic disease. And a paper a day comes out basically saying the same thing. Paper came out just 2 days ago documenting that sugared sweetened beverages was the primary driver of diabetes and heart disease around the world, especially in Pakistan.
Thanks for sharing that, this latest, paper, and we would love to go through that too. But the term which you've used, the druggable or foodable, I think that's a good take home for us, especially the people who consume this, as we call it, the standard American diet, which has come to our part of the world as well, actually all over the world. And so any comments on this food industry, which is pushing the SAD, the standard American diet?
Well, they need to, wake up and smell the coffee. The fact of the matter is that for the last 80 years, they have done very well. And they have done very well by providing a substance in our food that, number one, tastes good, and number two, is addicting.
Yep.
And they knew that. And that's why they use it. It's called sugar. And it doesn't matter if it's sucrose, table sugar, or high fructose corn syrup, or maple syrup, or honey, or agave. Each of those caloric sweeteners, those five, is one molecule of glucose, one molecule of fructose.
Mm-hmm.
Now, glucose is not the problem. Glucose is the energy of life.
Right.
Every cell on the planet burns glucose for energy. Glucose is so important that if you don't consume it, your body makes it. Okay? It's called gluconeogenesis. So it will turn glycerol from fatty acid breakdown into glucose, or it will turn amino acids into glucose. Okay, your liver will keep your blood glucose level up, even if you don't consume glucose. Carnivores consume no glucose. They still have a serum glucose level. The Inuit living in, the North Pole, okay, all they had was whale blubber and ice.
Mhm.
Okay, they still had a serum glucose level
Right.
because your body will take the fat and the protein and turn it into glucose. That doesn't mean you need to consume the glucose. So that's one of the problems of nutrition, because the dietitians will tell you, well, you need glucose to live. Well, yes, you do need glucose to live. You just don't have to eat it to live. Okay? You can do just fine without consuming
Right.
glucose. All right? But glucose is the energy of life, and every cell turns glucose into ATP.
Mm-hmm.
Every cell. All right? Now, fructose, this other molecule, this sweet molecule, okay, which is not glucose, even though the food industry tells you a sugar is a sugar, it is not.
Mm-hmm.
Okay, glucose is a six-membered ring, fructose is a five-membered ring. Okay, and their metabolic pathway within each cell, and within, specifically within the liver is where the real action is very different. Okay. And the reason is because fructose actually inhibits mitochondrial function. It inhibits 3 enzymes that are necessary for normal mitochondrial functioning. It inhibits AMP kinase, that fuel gauge on the liver cell we talked about before, directly, okay, by actually fitting inside the active site and preventing that AMP kinase from being able to actually bind an AMP, thereby turning it on. So it actually is a non-competitive inhibitor of AMP kinase by binding inside the active site in the gamma subunit. Number 2, it inhibits an enzyme called ACAD-L, acyl-CoA dehydrogenase long chain. This is an enzyme that's necessary to cleave fatty acids into 2 carbon fragments so that they can be burned inside the mitochondria. And finally, the third thing, fructose makes a lot of uric acid, and uric acid inhibits an enzyme in the mitochondria called CPT1, carnitine palmitoyl transferase 1. This is the enzyme that regenerates carnitine. And carnitine is necessary as the transport mechanism. It's the shuttle mechanism by which the fatty acids get out from outside the mitochondria to inside the mitochondria, past the mitochondrial membrane, so that those fatty acids ultimately can be oxidized into ATP. So in order to engage in fat oxidation and ATP generation. So the sum total of this is that fructose is a mitochondrial toxin. It causes mitochondria to be dysfunctional. And so when you consume sugar, instead of burning all the way to ATP, which is what your cell wants to do, what it's doing is it's inhibiting that. So now your cell is starving. So you get all the problems of cell starvation, fatigue, malaise, brain fog, not being able to, function like it should, weakness, sleep, sit on the couch, couch potato, etc. And also, you have basically diverted that energy metabolism off to growth. And so you will get glandular growth, like for instance, breast growth or prostate growth, increasing the risk for cancer. You will get vascular smooth muscle growth. So you will increase your coronary artery vascular smooth musculature and increase your risk for heart attacks. Okay. And ultimately, by laying down fat inside the cells, you are generating insulin resistance at the level of the liver. And so now you have hepatic insulin resistance. Now your pancreas has to make more insulin to make the liver do its job. So now you have hyperinsulinemia. Now you have an increased insulin burden all over the body, and the insulin's driving growth and driving cancer and driving heart disease. And ultimately, your pancreas burns out because it can't maintain that level of insulin release. And now you have insulin deficiency on top of insulin resistance, and now you have type 2 diabetes. So, fructose inhibition of mitochondrial function is at the nidus, at the heart of virtually every chronic metabolic disease that we know. And there's no medicine to fix that. The only thing to do is get rid of the substrate. And that means sugar reduction.
It's not druggable, it's foodable.
Foodable. That's right.
And so when we now move to, your famous line, which you are famous for, and we've heard about it. We've seen your podcasts. I've read your book. Protect your liver and feed your gut. Now, I feel that it is one of the most powerful lines when it comes to what we are doing in terms of creating health. And so please elaborate on that. So there's the 2 components: protect your liver, and the second is feed your gut.
Right. So protect your liver is basically so your liver mitochondria can work best. All right. So how do you do that? Well, If there's a toxin, take it away. That's the best way. All right. So what are the toxins to liver functioning? Well, fructose is the big one, and
Mm-hmm.
fructose is the one we could fix tomorrow. Fructose is the one that, basically every diet in the world that works, okay, is a low fructose diet. Okay. Pick your diet: keto, vegan, when it works, um, uh, traditional Japanese, uh, um, uh, uh, paleo, um, uh, Mediterranean, down the list. Okay. Every one of those is a low sugar diet.
Mm.
Okay.
Except
Oh,
sad.
and the one diet that doesn't work is the standard American diet, which is a high sugar diet. And the reason is because ultra processed food tastes like crap. And so what the food industry does is they add the sugar to hide all the off notes of
Mm-hmm.
all those ultra processed ingredients. Okay? Because you can make dog poop taste good with enough sugar. And basically that's what they're doing. So a standard American diet is a high sugar diet. Every diet around the world that works is a low sugar diet. And the
Mm.
reason is because fructose is a mitochondrial toxin.
Mm.
So getting rid of the sugar protects your liver. That's the first thing. Now there are other toxins that affect the liver as well. So heavy metals
Mm-hmm.
like cadmium and arsenic. Uh, there are chemicals in the environment that affect the liver, like chlorpyrifos and glyphosate, and many insecticides. Okay, um, there are obesogens in the environment that affect the liver, uh, chemicals that actually drive adiposity. Okay, and they affect the liver and cause the liver not to be able to process energy properly. Uh, like for instance, BPA and phthalates and uh, uh, PBDEs and many other
Right.
problematic chemicals, uh, which we've written about, in paper in Biochemical Pharmacology, 2022. So protect the liver. So protect the liver from toxins. That's the main
Yeah.
thing. Okay. Um, and then finally feed the gut. So why do you have to feed the gut? All right. Well, everyone now has heard of the microbiome.
Mm-hmm.
Okay. And they've also heard of this thing called microbial diversity.
Mm.
So there are some bacteria that are fragile and need sort of tender, loving care in order to be able to maintain their presence within the intestine. And there are some that are hardy and basically are impervious to virtually anything that happens to them. Perfect example of this is Clostridium difficile. Okay.
Mm.
You take a bunch of antibiotics, okay, and you kill off all the good bacteria, and the Clostridium difficile comes out instead, right?
Mm.
Okay, because it's very hardy. So the goal is don't let any one bacterium take over. Okay,
Mm-hmm.
you need a healthy mix so that no one bacterium can exert, a metabolic or a geographic advantage
Mm-hmm.
within the intestine over any other. Okay, and in order to do that, you basically have to, create the conditions for microbial health.
Mm.
All right. In order to do that, what do you have to do? Well, you have to give the bacteria the food they need to grow. Okay.
Yeah.
What is the food that the bacteria in the intestine need to grow? One word: fiber.
Okay.
Fiber is not food for you.
Mm.
Fiber is food for your microbiome. So you cannot digest fiber for energy. You cannot break the bonds in fiber, the beta glucans, et cetera. You don't have the enzymes to be able to do that, but the bacteria do. And so when you consume food with its inherent fiber, what you're doing is you're actually feeding your microbiome
Mm-hmm.
and you're increasing microbial diversity. Okay, so what is fiber? Fiber is a probiotic. So we
Okay.
have probiotics. Sorry, not probiotics. My mistake. Let's take that back. Fiber is a
Pre
prebiotic.
prebiotic, yeah.
Prebiotic. Okay. So we have prebiotics, which are food for the bacteria. We have probiotics, which are the bacteria themselves. And finally, we have postbiotics, which are the products that the bacteria make that are good for you. And there are people who are selling prebiotics, and there are people who are selling probiotics,
Hmm.
and there are people who are selling postbiotics, all in an attempt to try to get your gut, shall we say, healthy.
Right.
Right? Ultimately, real food supplies everything that your gut needs. It supplies the prebiotics, it supplies the probiotics, and it supplies the postbiotics. Okay, the postbiotics, by the way, come from fermented foods like
Yep.
kimchi, sauerkraut. The probiotics come from virtually anything you eat because, everything has bacteria in it. Yogurt as an example.
Hmm.
And the prebiotic is fiber. So you have yogurt, you have fiber, you have foods with bacteria in them, and then you have foods that have already been fermented. Okay.
Right.
Ultimately, those things improve gut health. And when you improve gut health, then you don't get the level of inflammation. And that inflammation causes mitochondrial dysfunction on its own. All right. So think of this way. Pretty much everyone, 93% of Americans today are inflamed. 93%. 93% of Americans have metabolic dysfunction due to gut inflammation. Okay. And the gut
Wow.
inflammation ultimately leads to systemic inflammation. So pretty much all of the inflammatory things that we are exposed to, not completely. I mean, air pollution is an inflammatory
Yeah.
agent and you get it through your lungs. Okay. Your skin for the most part, unless you put something on it, is relatively able to, maintain integrity. But the gut is where the action is. Remember, the
Hmm.
gut is where 1/2 of the immune cells in your body reside
Okay.
because your gut is your primary interactor between the outside world and your inside milieu, your internal milieu. Okay, the gut is the transducer of that. And so the immune cells are lining your gut throughout your small intestine, like Peyer's patches, etc. Okay, and think of it this way: your intestine is a sewer. Okay, what is a sewer? It's a pipe with you know what in it. Right? That's what a sewer is. Well, that's what your intestine is. It's a pipe with you know what in it. Now there are three barriers that prevent the you know what from getting into your bloodstream.
Hmm.
There's a physical barrier called the
Hmm.
mucin layer. There is a biochemical barrier, which are known as tight junctions. They are proteins that hold
Yeah.
the intestinal enterocytes together and so that they don't separate and allow stuff to get through. Okay. The most famous of which is zonulins. That's what goes wrong. That's what's defective in celiac disease. Okay. And then finally, you have an immunologic barrier. You have
Okay.
Th17 cells that secrete a cytokine called IL-17 to keep your gut uh, um, from being inflamed. Okay, so you have a physical barrier, you have a biochemical barrier, you have an immunologic barrier in
Hmm.
order to keep the junk in the lumen of the intestine and not into your bloodstream.
Hmm.
Okay, because inflammation is a mitochondrial toxin all by itself. Right? So, if you eat real food, you're maintaining those three barriers.
Hmm.
Once you switch to ultra processed food, the sugar in the ultra processed food causes the microbiome, okay, to chew up the fructose early, which means that the um, uh, uh, bacteria further down the intestine, they don't get their allotment. And so what happens is they're starving. They end up eating the mucin layer right off the surface of the intestinal epithelial cells. In addition, the emulsifiers in the ultra processed food that hold the fat and water together burn a hole in that mucin layer as well.
Hmm.
So you end up with destruction of that physical
Leaky gut.
barrier. And that's
As you
what
said.
leads to leaky gut.
Yeah.
Exactly right. And leaky gut means gut inflammation and then systemic inflammation. In addition, you have the enterocytes, the tight junctions holding the enterocytes together, those zonulins, et cetera. Well, that is an ATP dependent process. That is an
Mm-hmm.
energy dependent process. You have to create ATP in order to maintain the integrity and the function of those tight junctions. So anything that depletes ATP, even transiently, within those enterocytes can cause those tight junctions to become transiently dysfunctional and allow the junk to get through. Well, it turns out fructose, my favorite molecule, and others too, but fructose in particular, nitrates those tight junctions and renders them unavailable until a new one is made. And so you end up with transient dysfunction, and you can measure the endotoxinemia after a fructose meal. You can actually measure the lipopolysaccharides in the bloodstream. Sometimes you can actually measure whole bacteria getting
Mm-hmm.
across into the bloodstream. It's a very transient sepsis that occurs because from gram-negative organisms. So you know it's coming from the gut, okay, in response to a high sugar meal. So you know that the sugar is generating the inflammation because of the breakdown of those tight junctions.
Right.
And then finally, you have the immunologic barrier, those Th17 cells. So if you eat real food, those Th17 cells and the IL-17, they're all intact and everything's fine. If you eat a ketogenic diet, a high-fat diet, okay, they're still there. So you'll
Mm-hmm.
get more fat absorbed. That's true. But the barrier's still there. But if you eat fat and sugar together, which is, cafeteria diet, standard American diet, those Th17 cells are gone. They're replaced by Th1 cells, and the IL-17 is gone. And now basically all hell breaks loose. And you end up with, immunologic deficiency within the intestine, and therefore the ability of pathogens to be able to broach the barrier. So every single one of the barriers is made worse by
Mm-hmm.
sugar. Okay. There are other
Specifically
things
fructose.
too. Right. There are other things too, like for instance, the lack of fiber and also emulsifiers. Okay. All these things that are in ultra processed food. But sugar's, by far and away the worst. And then there's the omega 3s, which we, sort of just touched on briefly. Omega 3s are actually what are used for mitochondrial structure. So
Mm-hmm.
they're actually contributing to the maintenance and the synthesis of new mitochondria. So when you are omega 3 deficient, okay, that's going to put your mitochondria at a disadvantage. And so you're not going to be able to do as well. We also have learned that when you're omega 3 replete, okay, you will make fewer reactive oxygen species out of your
Mm-hmm.
mitochondria. Your mitochondria make reactive oxygen species as a matter of course. That's just part of, the reactions of life. Okay. But normally those are quenched by antioxidants in the peroxisomes of the cell. But if you're making too many, you may overwhelm your ability to be able to quench those. And then those reactive oxygen species will cause damage as well. So if you have defective mitochondria because you are omega 3 deficient, you're going to generate more reactive oxygen species and you're not going to be able to keep up. And that is also going to cause metabolic damage. So, high sugar, low fiber, low omega 3s, high emulsifiers.
Yeah.
Okay?
Yeah. Makes sense.
That's ultra processed food.
Yeah.
Now, there are other things
And
wrong with ultra processed food too, like the colorings and what have you. But those four things are what contribute to chronic metabolic disease. And none of those, by the way, are captured in the concept of nutrition.
got it. So, so you mentioned real food. What do you mean by that?
Very simple. Food that came out of the ground or animals that ate the food that came out of the ground. That's real food.
Okay.
Okay. As soon as it went to a packaging plant or processing plant for something to be done to it,
Mm.
it's processed. Now, sometimes processing is good, like for instance, pasteurization.
Mm-hmm.
Okay. Um, sometimes processing can be not harmful, like basically taking the skin off an apple, and selling apple slices. That's not a big deal. Right? It's when you start getting down to the components, the individual components of food, okay, and stripping away the matrix that the food is in, which is for the most part fiber, okay, and start dumping in all sorts of things that didn't belong there. That's when you get into problems. And that's basically the definition of ultra processed food. So, how do you tell whether something is ultra processed or not? Um, our colleague, Dr. Carlos Montero at the University of Sao Paulo, did us all a very great service. He developed a system of food classification, and it's called the NOVA system, NOVA. And what the NOVA system does is it divides all foods into four categories.
Mm-hmm.
And the easiest way to explain this would be an example. Let's take an apple.
Right.
So Nova Class 1 would be an apple picked off a tree.
Mm-hmm.
Nova Class 2 would be apple slices, destemmed, deseeded, deskinned, maybe. Nova Class 3 would be apple sauce. So macerated, pulverized, possibly a preservative added, maybe a little extra sugar.
Mm-hmm.
Nova Class 4 would be a McDonald's apple pie. Now, when you actually look at the nutritional epidemiologic data, it's only that Nova Class 4 group, that ultra processed group, that contributes to disease. And
Okay.
Carlos has done a very good job
Yeah.
all
Yeah,
over
I've
the
seen that.
world demonstrating that it's the ultra processed food category that leads to the disease. And it's type 2 diabetes and hypertension and dyslipidemia and cardiovascular disease and cancer and dementia, fatty liver disease, polycystic ovarian disease, and also attention deficit disorder, depression. I mean, it's a lot of stuff. And the reason is because of mitochondrial dysfunction
Yeah.
due to ultra processed food consumption. And when you fix that, those diseases reverse, they get better.
Yeah. So
So
you
ultra
spoke a
processed
little
food
bit
is
about, I'm sorry, you spoke a little bit about, these diets, the vegan or the ketogenic or the Mediterranean. So which diet do you think is better? Are all these diets better than the SAD?
all of them are better than the standard American diet. The standard American diet is by far and away the worst diet in the history of mankind because it's not really food.
Mm.
Not really food. I mean, think of it this way. Okay. What is the definition of food? What is the definition of food? If you go to the dictionary, what does it say is the definition of food? Substrate that contributes to either growth or burning of an organism.
Right.
That is the definition of food. I completely agree. 150% agree with that definition.
Mm-hmm.
Okay, so let's look at that. Does sugar stimulate burning? I just showed you that sugar inhibits three enzymes necessary for mitochondrial function. In fact, sugar inhibits burning. And
Mm.
virtually all ultra processed foods are high sugar on purpose.
Right.
Okay. Does ultra processed foods stimulate growth? Well, my colleague, Dr. Efrat Monsonigo Ornan at Hebrew University, Jerusalem, studied this and actually looked at long bone growth, trabecular bone growth, cancellous bone growth, cortical bone growth as a function of ultra processed food and sugar. And it
Mm-hmm.
turns out that ultra processed food actually inhibits bone growth.
Okay.
It inhibits bone strength. It inhibits tensile capability, to keep from being fractured. Okay, the lacunae are all hollowed out. It's a disaster. It actually
Mm-hmm.
inhibits growth, and of course, because of the inhibition of mitochondria, it actually stimulates cancer growth instead. So it hijacks growth. So you don't get the growth you want, you get the growth you don't. So if a substance that passes your lips, if a consumable does not stimulate growth and does not stimulate burning,
Mm.
is it a food?
Then it's not food.
Then it's not food. Then it's a poison.
Mm. Yeah,
Ultra processed food
that's a very good
is
definition, and the
poison.
way to present that. I just wanted you to maybe a short comment on non-alcoholic fatty liver disease,
Sure.
which we have more than 45% of the American population has it, and I
Right.
and I heard you that
And
even children
30% of the
has it.
Pakistani and 30% of the Pakistani population has it.
Yeah,
So this is a huge problem. And actually, I had a slide, if you want to see that let me see if I
please.
can I show?
Yeah, yeah, sure. Go ahead, please.
All right, let me find it for you. All right, it just
And
came.
NAFLD is only because of fructose?
No, no, no. Anything that inhibits liver mitochondrial function will increase NAFLD. Here we go, NAFLD in Pakistan. Here we go, right here. Okay, so here's NAFLD in the general adult population over here on the left. And you can see it's 29.82% of the general adult population
Mm-hmm.
in Pakistan, which is insane. Okay. I mean, the only country that's worse is America, where it's
Right.
45%. All right. And more importantly, this is the prevalence of NAFLD in patients with diabetes in Pakistan, 58%.
Mm.
And in fact, the reason is because fatty liver is a precursor to diabetes.
Mm.
Once you get fatty liver, you are 3.5 times more likely to develop diabetes because of your fatty liver, because your liver doesn't work right. And if your liver doesn't work right, then your pancreas has to make more insulin.
Yeah.
And if your pancreas has to make more insulin, it'll burn out. By the way, we did a study of children where we took the sugar out of their diet and we measured their fat. And we very specifically measured their liver fat, but we
Mm-hmm.
also measured the fat in their pancreas.
Right.
And it turned out these kids had fat in their pancreas. And when we took the sugar out of their diet and replaced it with starch, so we took the fructose out and we replaced it with glucose, the fat in their pancreas disappeared too.
And and
Fat,
and how much time did it disappear?
10 days,
Wow.
10 days. So in fact, the diabetes may be non-alcoholic fatty liver disease and non-alcoholic fatty pancreas disease.
Mm.
Not just NAFLD, but NAFPUD. And so this is extraordinarily important in
Yeah.
terms of uh, understanding the nature of chronic metabolic disease. You do not want fat in your liver. You
Yeah.
do
I'm
not
I'm glad
want
I
fat in
asked
your
this
liver
question.
or pancreas.
Yeah. I'm really glad that I asked this question. So now we're actually moving towards the end of our conversation. Uh, I just had a few more questions. So we have 10 minutes. And so I just wanted to understand your take. And I've heard you talk a lot about, calories and the glycemic index. Maybe a little bit on that.
Well, so the dietitians, they love this concept called glycemic index.
Mm,
I don't.
mm,
So why do they love it and I don't? So glycemic index, what is it? It's how high will your blood glucose rise after 50 grams of carbohydrate in a given food.
okay.
Now, the size of the glucose rise, the amplitude of the glucose rise, is a proxy for the amplitude of the insulin rise,
Hmm.
because the higher the glucose, the higher the insulin. So instead of measuring the insulin response, which is really what you want to know, they're measuring the glucose response as a proxy. Okay. Now, that's not terrible because the glucose rise and the insulin rise are correlated with each other.
Mm-hmm.
That's true. So that's not the terrible part. That's not why I'm against it. Okay. The reason I'm against it is because there are two problems with it. Okay. The first problem is fiber.
Okay.
So the best way to explain this is carrots. So carrots, if you eat 50 grams of carbohydrate in carrots, your blood glucose will go pretty high.
Mm-hmm.
So carrots have a high glycemic index. The question is, how many carrots do you have to eat to get the 50 grams?
Yeah.
Turns out you have to eat 700 grams of carrots to
Yeah.
get 50 grams of carbohydrate. Okay? Because the entire carrot is fiber.
Yeah.
Okay? It's all fiber. So glycemic index doesn't take fiber into account. Is there something that does? Glycemic load. So
Okay.
glycemic load is how many carrots do you have to eat to get the 50 grams. So carrots are high glycemic index, low glycemic load.
Mm-hmm.
Carrots are good. But if you listen to the dietitian, carrots are bad. So that's problem number one with glycemic index.
Mm-hmm.
Problem number two with glycemic index, fructose. So fructose does
Yeah.
not raise the blood glucose. Fructose raises the blood fructose. You don't measure the fructose in the blood glucose.
And can you
Fructose
measure
is,
that?
well, you can, but it's a research tool. It's only
Okay.
done in certain labs. You can't do it routinely. Okay. I'm hoping that in the future we will be able to do it, but at the moment we can't because
Right.
fructose and glucose are both the same size. They're both the same molecular weight. They both are the same charge. So they're very hard to separate from each other. But it is doable. It's just not doable routinely.
Okay.
The point is, fructose is 7 times worse at generating reactive oxygen species, at inhibiting mitochondria, et cetera. Okay, so the amount of fructose matters a lot, but it's not measured in the concept of glycemic index.
Yeah. Makes
So
sense.
So there are two problems with glycemic index, and the dietitians who are proposing and espousing glycemic index, they don't deal with either of them because they don't know how. So I hate glycemic index. I think glycemic index needs to be thrown in the garbage can, okay, as a concept. What we care about is the insulin response. That we
Okay.
do care about. Now, can you learn that? Can you measure that? Yes, absolutely. But it's a little bit more complicated.
Right. So a few quick questions. So which are the tests we should go for to evaluate our metabolic health?
So there are a lot of tests. and the question is, which ones give you the most value?
Right.
That's really what you want. Okay. And which ones are the cheapest?
Mm-hmm.
Okay. So the best test is the most expensive test. Okay. Which would be a magnetic resonance spectroscopy of your liver.
Okay.
Okay. But you don't carry an MRI machine in your back pocket, and your insurance company's not going to pay for that, et cetera.
Mm.
So that's not really a very good test if you can't get it. It's a
Mm.
great test in terms of accuracy. It's a lousy test in terms of feasibility, scalability, and
Any lab tests?
clinical use. So now let's talk about lab tests, right?
Mm.
So the lab tests are the following. First of all, the cheapest is the one that's free, and it's a waist circumference.
Okay.
Because
Yeah.
your liver fat and your waist circumference are usually correlated. So if your waist circumference is going up, there's a good chance your liver fat's going up too. So
Mm.
that's free. Okay. The problem with it is inter-observer variability and
Yeah.
getting too close to the patient, and sometimes they
And
smell bad.
I think it varies from uh, countries and
Yes, absolutely. It's age and sex and race dependent,
yeah.
and the markers are different.
But still, it's cheap. It's available
Right.
and still it can
You
give
should
us some information. Yeah.
you should
So,
you should do it because it's free
mm,
and it does give information. So now let's talk about the blood tests.
yeah,
So the best blood test is a fasting insulin
fasting
by far and
insulin.
away. Fasting insulin.
Okay.
Fasting, but it's got to be fasting. If it's not fasting, don't bother.
Mm-hmm.
Got to be fasting. And a lot of patients, you know, you tell them to come in fasting and they forget. Okay. And they ate, you know, they had, coffee with their, milk and sugar
Yeah.
before they got there, because they can't, get to the lab before that, so that's a problem. But a fasting insulin tells you the most. And the reason is because a fasting insulin is immediately downstream of mitochondrial dysfunction.
Mm,
So when your mitochondria don't work, okay, that tells your pancreas you have to make more insulin to try to make your
mm-hmm.
mitochondria work, and it doesn't. So,
Wow.
so a fasting insulin is the best indirect arbiter of mitochondrial dysfunction. Now, the American Diabetes Association says don't draw a fasting insulin. I say it's the most important test you can do. So
Mm.
how come they say don't draw it and I say it's the most important? Because I'm right and they're wrong. That's why. But why? Why am I right? And why are they wrong?
Yeah.
Why?
Interesting.
So they say it for two reasons. The first reason they say don't draw it is fasting insulin assays are not standardized across the various platforms.
Mm,
So there are some expensive tests for fasting insulin, and there's some cheap ones. The cheap ones tend to be radioimmunoassays or ELISAs, and cross-reacting substances can be picked up in that assay. The most important of which is proinsulin. So proinsulin is like insulin, but not. It is the precursor to insulin. It's
mm-hmm.
made by the beta cell. Okay, you make proinsulin, which is one protein, one peptide, and then an enzyme comes along called prohormone convertase 1, and it cleaves a piece out of that protein, and we call that C-peptide,
Mm-hmm.
okay, which you can measure. And then you have the mature insulin, and the mature insulin is way more powerful at glucose disposal and metabolic health than proinsulin is.
Mm-hmm.
Proinsulin has 5% of the activity of insulin, but proinsulin is measured in the insulin assay. So if your beta cells are under uh, fire and they're working really hard, they will put out proinsulin, which is not insulin, but you'll
Mm.
measure it in the insulin assay. And so the insulin level will look high, but you're not measuring insulin, you're measuring proinsulin. So that's one of the big problems. To me, and that's true, I say, so what? If it's high, you got a problem. Okay? The second reason that the ADA says don't draw it is because fasting insulin levels do not correlate with obesity. That's right, they don't. They correlate with metabolic health.
Yeah.
And there are plenty of people in Pakistan who are metabolically unhealthy but thin. You can be thin sick, and you can also be fat healthy for that matter.
Yeah. You use the term fat sick people and,
Well,
metabolically healthy
right,
obese.
right. Metabolically healthy obese, and also this other thing called TOFI, T-O-F-I, thin on the outside, fat on the inside.
Yeah.
Okay. And a lot of people in Pakistan are. So fasting insulin will answer that. So that's why I think fasting insulin is the most important
Right.
test you can do. What other tests? Well, an ALT, alanine
Mm-hmm.
aminotransferase. But in order to interpret an ALT, you have to know the normal range. And the problem is the normal range on the lab slip is not the normal range. Because the normal range for ALT used to be up to 25, and now the normal range for ALT is up to 40 on the lab
Mm-hmm.
slip. So how did it go from 25 to 40? Because everyone has fatty liver disease.
Yeah.
And how do you decide a normal range? Okay, you take 10,000 people who say they're healthy and you measure them, their ALT, okay, and you construct a Gaussian distribution and you do, two standard deviations from the mean and you do a cutoff there and you say, okay, that's the normal range. Well, if the
Yeah. That's
entire curve
shifted.
is shifted to the right,
Yeah.
okay, you got a problem. Okay. So you have to use the right normal range, which is from 50 years ago before people got sick. All right. So the ALT, another one is uric acid. And
Okay.
uric acid is necessary because uric acid is an inhibitor of mitochondrial function, and it's a manifestation of increased fructose and also purine consumption. Okay. And then finally, ApoB, which is a combination of LDL and triglyceride.
Okay.
So those are the things that I say are the most important that you can measure on a
Right.
lab slip.
Thank you. I think that's a good list. So a few questions. So one, just a short one. Can chronic diseases be reversed by food and lifestyle changes?
Can they be changed?
Reversed?
Reversed, I'm sorry. Absolutely. Almost all chronic diseases can be reversed if you fix the problem. People,
Okay.
doctors will tell you that type 2 diabetes is a chronic, unremitting, unrelenting, progressive, problem that never gets better. Garbage. Completely untrue. All of those things are true if you don't fix your diet.
Yeah.
But if
So
you
it's
fix your
foodable.
diet, you can reverse
Yeah,
it.
the
It's
foodable
foodable.
term, I think we'll take that. And what's your take on, as I mentioned, lifestyle medicine, integrative and functional medicine?
So, in general, first of all, I'm not an integrative or functional medicine doctor. I'm an
Mm,
endocrinologist, ivory tower, UCSF endocrinologist. So I'm not here to, promote or, dissuade people from going to integrative medicine or functional medicine doctors.
right.
What I am saying is that medicine has become very siloed. Okay. Each of the specialties in medicine have basically taken their organ and figured out what's going on with their organ.
Yeah.
Chronic metabolic disease is a systems biology issue. You have to think of it in systems biological terms. Mitochondria is a systems biology problem because every cell in your body has mitochondria, needs mitochondria to work. When your mitochondria are defective, the disease you get is whatever organ the mitochondria are in that have the problem. So if the mitochondria are sick in your ovary, you have polycystic ovarian disease. If the mitochondria in your body are sick in your heart, you have heart failure.
Yeah.
If the mitochondria in your liver are sick, you have fatty liver disease, etc. The only way to solve this is by integrating all of this into a systems biological perspective.
Mm,
Integrative medicine tries to do that.
yeah.
Functional medicine tries to do that. Functional medicine tries to fix it with things that they have available to them, like supplements. I think real food will solve most of these. So
Okay.
I'm not for or against integrative medicine or functional medicine. I'm for real food.
Got it. Okay. I think that, yeah, this is great. This 1 hour has, and I'm sure that, this will really, really help. As I said at the outset, this will really change the mindset. And this, if I may call this a narrative, which is obviously evidence-based and goes down, deep down to the cellular level. I think this is a game-changing concept. So thank you very much. Before I close, I would like to share two quotations, which I always, mention in my talks as well. And the first is from Sir William Osler, who's the father of modern medicine. And he said this around 100 years ago. One of the first duties of the physician is to educate the masses not to take medicine. That's your druggable,
Yep.
statement. So just
Right.
wanted to add to that. And
Mhm.
then Thomas Edison said that the doctor of the future will give no medication, but will interest his patients in the care of the human frame, diet, and in the cause and prevention of disease.
Wow.
I think that's what we have elaborated today. Uh, before I thank you, any one-liner, a take-home, adding to what you've already said?
You can't fix a problem if you don't know what the problem is. Okay. What I've hope I've done in the last hour is shown you what the problem is.
Yeah,
Then
you
we can
certainly
fix it.
have.
Then we can
And so I thank you, Rob. I really thank you for educating us and opening our minds with the knowledge which you have shared in such a short time. One of the most comprehensive conversations I've ever had. I look forward to having more deeper conversations with you to give a more comprehensive understanding of insulin resistance. I think we need to have a very detailed conversation on that and how to achieve metabolic health and complete wellness, which we all deserve. So
Indeed we do.
thank you very much.
You're very
I
welcome.
really appreciate your time and really look forward to seeing you again very soon so that we can have the next conversation building upon what we have created. So thank you very much and have a great day.